Yeah. Okay, I've turned on the recording. Okay. The recording is on now, right?
Yes, sir.
Okay.
Yes, sir.
So let us imagine that this is a chromosome, okay? And you got to know the sequence of only this part somehow. Okay?
So that blank slide is
Are
not
you
visible.
drawing anything?
You are not. Are you not seeing it?
No, sir.
So
Okay.
startup
No, sir.
slide is there.
Okay. Can you see it now?
Yes, sir.
Yes, sir.
Okay. So now imagine that this black thing is the whole chromosome, and somehow you got to know the sequence only corresponding to the pink part. Okay? Okay?
Yes, sir.
Yes, sir. Yes, sir.
Now, once you get to know the sequence of that part, then because you know the sequence of that part, you should be able to design two primers, right? One from here, one from here, right?
Yeah.
Yes, sir.
Yes, sir.
And these primers can extend in this way and this way, right?
Yes, sir.
In the next round, then you will get to know the sequence of this. Then you again go back and design primers for this part and for this part, so that it can read outside, it can read outside. This way you, like, this is
Sir,
how
we can also do the ligation into a ligation and then primer so that it will synthesize.
it is the more or less the same thing that
Yes.
you are talking about, that if you ligate it and then you design two primers, one reading in this direction, another the reading is this direction. It's more or less the same thing.
Okay.
So this is how Francis Collins was attempting to sequence the whole human genome.
Okay.
Okay, like the upper part.
So here, like, this way we are just replicating those genes now. I mean, from where this sequencing is coming.
I did not understand.
So here, what are we doing? Like, we are adding the primers and replicating the gene already available now.
No, absolutely not. Absolutely not. You are not, like, if you consider sequencing as replication, then yes, but you are not replicating, you are only sequencing. So you will take the whole chromosome and then you will, cut the DNA, like by sonication or something like that, into small pieces, and you will incubate it with the primer and you will do the sequencing. And because you know the orientation of the primer, you know that the sequencing will move away from the known sequence. So you know the immediately neighboring area.
Okay, yeah.
You don't have to replicate or anything.
Yeah.
If you have any confusion, please feel free to ask more questions.
Sir, the last portion I did not understood. Without the replication, how should we know the sequence of the neighboring portions?
Like, first let me get one thing clear. Sequencing by definition is a form of replication. Do you mean that way, or do you mean actual replication of the DNA?
Okay, sir, I meant about the replication to know about the sequence. So not the second one, first one.
That is always true, right? Sequencing is always dependent on replication only.
Okay.
Like, to be scientifically correct, the language to be used is sequencing is dependent on DNA-dependent DNA polymerization.
Okay, sir.
Are we using DDTs to stop the replication for, I mean, to
You will,
wherever?
Of course, you will use DD-NTPs that are differentially fluorescently tagged.
Oh, okay. Then yeah.
And Navneet?
Yes, sir. So I have a question about the cloning experiment we did yesterday
Uh-huh.
in yesterday's class. So in that we actually had the KPN1 site in the forward primer
Uh-huh.
and SAC site in the reverse primer.
Mm-hmm.
So if I'm correct, I worked it out, and if you clone it inside the P blue script plasmid, it's actually the T3 promoter is actually upstream to the 5' end of the antisense strand. So if that's the case,
Mm-hmm.
then the transcript will be antisense transcript and not the sense transcript. So
Yes.
that was my question.
Yeah, but in P blue script, if you look at it carefully, then on the KPN side, you will see T7 promoter. So if you want to make the sense transcript, you can use the T7 promoter or polymerase. If you want to make the antisense transcript, you can use the T3 polymerase. So if you search the literature, you will find something called coupled transcription translation system. Okay, if we like say, like, forget about now, in the olden days, or even now, if you discover a new organism, you want to ensure that the clone that you have made is actually the full length clone. Like, you don't want to leave anything to chance. Okay, so the way it was done is that you will have a tube. Yeah, you will have a tube. Within that, you will put your plasmid in P-blue script, and you will use the promoter, like either the T3 or T7, whichever will make the mRNA equivalent RNA transcript. And in the same thing, you will also add an in vitro translation system, okay, which will have ribosome and ATP regeneration system and everything, and S35 labeled ATP. So you will make the only protein that will be radioactive is the one which is being transcribed from this plasmid and the RNA being translated. So then you run that on a gel, and it will migrate with the size, and you already, because you have discovered the clone after knowing after full characterization of the protein. So if the naturally occurring protein that you have purified and your in vitro transcribed translated one is the same size, then you are confident that you have a full length clone. So yeah, you can there are cases where we make the sense transcript. There are cases where we make the antisense transcript, depending on the application.
Okay. And where would we use the antisense transcript? Like, it doesn't code for any protein, right?
Yeah, but for RNA in situ hybridization.
Okay. All right. And one more question, that like the host organism, the choice of host organism varies, is it? So if we are looking for T3 RNA polymerase, it's a different host organism, and if it's
No,
T7, it's
no, no,
different.
no, 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 absolutely not. This in vitro transcription has nothing to do with host. You will have a tube.
No, I actually meant the cloning the plasmid inside E. coli or some other bacteria.
No, no, you clone the plasmid inside you clone the DNA inside a plasmid. Now you decide which host to put it in. Now that will depend on the primary thing, whether you want to express the protein corresponding to the DNA, or you want to just clone it for further use. Because expression plasmids, generally speaking, they are not very high copy number plasmids. So they are not the plasmid of choice if you want to make a lot of DNA corresponding to your particular clone. Okay, so now you tell me, now you ask your question again to me. What is it that you are trying to do?
Okay, so if we are looking for expression,
If you are looking for expression, then you have to go to a different plasmid altogether. We will do that after the quiz. Okay,
okay,
I will upload those lectures during the weekend.
so like I have just one question. So if we are looking for molecular cloning inside the host, so in that case, does the orientation really matter when we clone?
Yes, yes, it may matter. It depends on what is it that you want to do, what is it that you have in your laboratory, etc., etc. If you are trying to make antisense RNA, and for some reason your lab only has T3 RNA polymerase, then you will ensure that the gene is cloned in such a manner that the 3' end is towards the T3 RNA polymerase.
Okay.
The T3 promoter. But in those cases, you isolate the plasmid and make the transcript in a test tube. Like you will have a tube. You will have a test tube. There you will have the plasmid. Then you will separately add RNA pol. It can be T3, it can be T7, depending on what you want. And then the RNA will be made in vitro. Okay. Navneet, do you understand?
Yes, I got it. Thank you.
Okay, Ayush.
Sir, can you please go back to the first slide?
Yeah.
So in this slide, as we can see in the lower pink one, we are using primers from the two ends, and they are going like, I cannot define the directionality as there is no 5' to 3',
Yeah.
but they are going in the opposite direction. They are synthesizing. So basically, they do not have any limit to which up to which they will extend the
yes.
So how can we define that the length of whichever fragment they are synthesizing will be equal to the one which is on the upper side of this?
No, you do not have to, right? Let us say that in this direction, it gave you a sequence of 700 base pair, and in this direction, it gave you a sequence of 500 base pair. That's fine. You got to know the sequence of 1200 base pairs. Then you will design new primers, say around 450 base pair here, 650 base pair here. Then again, it will extend for another 500, 600 bases. So that's how you will get to know.
So that actually depends upon the fidelity of whatever enzyme that we are using.
Yeah. Fidelity, luck.
Okay.
Yeah.
Okay. And can you please also explain RNA-seq technique, because I was not very clear about it during the lecture.
Okay, so that is actually RNA-seq is just NGS combined with preparation of cDNA library. Okay, so you will have the RNA, right? Now you make your first strand cDNA. Now you couple it with like if you want, you can make the double strand. I do not know exactly how they make it in the industry, but technically speaking, you can make the double stranded cDNA like by ligating with the G adapter, and then doing the second strand synthesis. You can make the double stranded cDNA, right?
Yes, sir.
Yes, sir.
And then you can digest it with some restriction enzymes like I'm forgetting, like typical tetrahedral restriction enzymes, that are a tetracutter will have what? 256 nucleotides?
Four.
Like,
Yes, sir. Yes, sir.
Yes, sir.
so you will have typically fragments of the size of around 256 nucleotides, and then you ligate it with the adapter for the NGS platform, and then you immobilize it on the slide, and then you do your sequence just the way you were sequencing genomic DNA.
Okay, so the sequencing is basically taking place by the principle of synthesis rather than ligation, right?
Yeah.
Okay, okay. Thank you so much, sir.
Sure.
So can we move into microarray now?
Yeah.
So the first question from microarray is like, you have told about the 60 MERS, 60 oligonucleotide probes.
Yeah.
Um,
Not oligonucleotide probes, okay? What is
Okay.
there on the array, those are not called probes.
Okay.
Those are your templates.
Okay, those are my templates.
What you add from outside is the probe.
Okay, so means like from I want to know like which genes are responsible for some pathway in muscle cells. So
Okay.
I have extracted the total mRNA and converted it into the cDNA or whatever. So that will be my probe.
Yes.
Okay.
Because you will add the fluorescence at that time during the synthesis of the cDNA.
Okay. So that will be my probe, and what is there in on the array will be my template.
Yeah.
Okay, so this doubt was there. Okay.
Yeah. So, the way I like to project it is that what you have on the array is your superset.
Okay.
And now, like, it is supposed to represent every mRNA that can possibly be expressed in a human being.
Yes, all the 23,000 genes.
Yes.
Yeah.
Now, your muscle-specific genes is a subset of it.
Yes.
And your, kidney-specific genes is another subset of it with some overlap. So that's basically what microarray is all about.
Okay, sir.
Any other question?
Sir, can you explain the concept of internal controls and the means positive and negative controls?
Yes. On the array or in the probe? Let us do both. Let us do both.
Yes, sir. Yes.
Okay. So let us first talk about on the array. Okay. So end of the day, it's a hybridization based technique, right?
Yes.
So some infidelity may be there in hybridization. Non-specific hybridizations are known to happen. Okay. The way we never say that we have eliminated non-specific hybridization. We say we have minimized non-specific hybridization. So how do you minimize it? How do you know that you have minimized it? So if this is my array last slide, imagine that in this quadrant, I have put only sequences that are unique to East. You all understood that how you can get unique 60 MER for each mRNA, right?
Yes, sir.
Okay. If then you will similarly be able to get unique 60 MERs for East. Let us say that this quadrant is for East. Now,
so unique 60 MERs of East means they will be present in all the cells of, or what? This was not clear, sir.
तब तो कुछ भी clear नहीं हुआ. See, every mRNA has a sequence, right? Now that particular mRNA I am purposefully not getting into the complexity of splice variants right now. But if we imagine that, there are 23,000 different mRNAs, no matter which cell the mRNA is produced, it will have the same sequence, right?
Yeah.
Now, you can, like the gene pathway that I worked with, signaling pathway, is called BMP signaling pathway. Okay, there are 20 different BMP ligands. Okay, so now you understand during evolution they have been duplicated, right? You would expect very high sequence homology between all BMPs.
Yes.
At least at the amino acid level.
Yeah.
So now there is a possibility that at nucleotide level also there will be quite significant homology. Now, if you want to know whether in a particular manipulation, whether it is BMP2 that is going up or BMP4 that is going up, if you are using a probe that does not distinguish, like if you are using a template sequence that cannot distinguish between these two, then your purpose is defeated, right?
Yes.
So if you align all the sequences of mRNA sequences, because please understand that these are post-genome sequence era technology. Today, you know the sequences of every single mRNA, right?
Yes.
So if you do a massive scale alignment, you will be able to find a tiny stretch. I am marking it by thick yellow line. Which is not present in any other mRNA, right?
Yeah.
By sequence alignment. So that this yellow patch, if it is 60 nucleotide long, so you know a stretch of 60 nucleotide that is exclusive for BMP2. Another 60 nucleotide that is exclusive for BMP4. Exclusive meaning in none of the 23,000 mRNAs it will be there.
Okay, so that 60 nucleotide stretch is unique to only one mRNA.
Yes,
Exclusively.
one mRNA and only one mRNA.
Yes, exclusively. Okay.
Okay, and the
Yes.
the reason they do it 60 mar is that the probability of random occurrence of a stretch of 60 mar exactly is very, very low. Right? Now, these are not like, these are actually sequences. You know the ATGC of this 60, right?
Yes.
You can order your inkjet printer lithography machine to that on this spot you make the 60 mar for BMP2, on this spot you make the 60 mar for BMP4, on this spot you make the 60 mar for INT6, so on and so forth. You can actually print 40,000 spots, right?
Yes.
Now you also print take an area. I am not saying it has to be secluded, it can be intermingled, it doesn't matter, which will be 60 mars from east mRNA. Now, if you are trying to say you are trying to know the genes.
Yeah, so it will act as a negative control.
Yeah, so let me explain that to you. So say you are trying to identify the genes that are transcribed right after BMP ligand binds its receptor. Okay, so you take a cell line that has BMP receptor, you add commercially available, sorry, can you see me, hear me now?
Yes.
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 From this morning, my net is creating a lot of trouble. So once the ligand binds to the receptor, there will be some immediate early transcriptional changes, right? So if there is another cell, if there is another cell which also has the same receptor, but in this cell you did not add the ligand, so the mRNAs that will be produced in here, those are not likely to be produced there, right?
Yes.
So now you make cDNA in here and cDNA in the other cell as well. You mark these with green, and you mark these ones with red. Okay, and then you hybridize it to this array. Now to this array. Now what will happen is that these are both mRNAs from human cell, right?
Yes.
So none of those should be able to hybridize anywhere in this quadrant where East mRNA like templates are there.
Yes.
So if you see that in your hybridization microarray, none of the East the templates have lighted up, so you know that your hybridization condition is stringent enough that there is no non-specific hybridization.
I have minimized the non-specific hybridization.
Now you also do something else, that you ensure that on your slide there are I am drawing BMP 2 mRNA with a different color just for the illustration purpose. That you decide that you will have 5 different spots for the same 60-mer of BMP2 mRNA. Okay. So now, if in this case BMP2 mRNA is one of the early responsive transcript, so that should hybridize to all the spots of BMP2.
Yes.
Not just one. So that is the basis of algorithm. That are we getting signal from all the spots that are in duplicate? Are we not getting any signal in the non-specific area? Based on that, they have complex algorithm. I do not know exactly how the algorithm is done, but this is the operating principle.
Yes, sir.
Is the recording still on?
Yes, sir.
Yes, sir.
Thank you. Anything else? Yeah, Shreya, bolo.
Sir, for microarray analysis, like, are we only doing qualitative analysis or quantitative analysis, or both?
Very good question. Very, very good question. Shreya, what is your background?
So I have done MSc in biotechnology.
वहीं पे गड़बड़ कर दिया. So, have you ever done any blue-white selection?
No, sir.
Okay. Have you ever done any chemical reaction that produces color?
Yes, sir. Like for ELISA, I have like done.
अच्छा, ELISA के पहले कुछ नहीं किए हो? Like very ordinary color reactions, say Biuret method and stuff like that.
Yes, sir.
The protein estimation techniques.
Yes, sir. Yes,
It's
sir.
the Laurie's method.
Yeah, Laurie's method.
So when you produce color,
Yes, sir.
what happens? Like in all these reactions, when you are producing color, what happens? Do you know? I'll give you the answer. The color is actually precipitated. It's a colored precipitate that forms, right?
Yes, sir.
Right or not?
Yes, sir.
Yes, sir.
Okay. So now, if you allow the reaction to happen for long enough, what will happen? How will the graph go?
So more and more
So,
precipitate will be.
so initially it will like increase, and then after certain point it will saturate,
Why will it saturate?
because there will be no more substrate molecules available to get it
Okay,
converted to
I
the
will supply
color.
you, I will supply you more substrate. Then what will happen?
Uh, so the product which is already formed can inhibit the formation of more product. Like,
No, when you are forming a precipitate, it is taken out of the system. It will not inhibit. See, what happens is that we refer to this as something called saturation kinetics. It saturates, like the intensity of the color change. Like it does not mean that the precipitate formation is being stopped. Like what you were saying, Shreya, that also happens. Okay, for reversible chemical like enzymatic reactions, what you were saying is the correct thing. But when you were talking about this, any reaction that results in a precipitate, and you are detecting the amount of precipitate, whether perimetrically or otherwise doesn't matter, then beyond the threshold, the color intensity does not change. Or even if it changes, it does not change linearly.
okay.
Okay. Now you think of microarray. Okay.
Okay.
It is a method based on hybridization, right?
Yes.
Here it will be only those many number of probes will be able to hybridize as many templates are available on your array.
Right.
So if the maximum, I am making up these numbers, if the maximum number of probes that can possibly hybridize for a particular species on the array is 10, then if the abundance is between 0 and 10, you will get quantitative output. But if the abundance varies from 0 to 100, then of 10, whether it is 11 or 99, it will not make any difference, right?
Yeah.
So that is the reason why microarray is not a strictly quantitative method. It is a qualitative method. It gives you some idea. Like you cannot make a lot of noise based on fold change in signal intensity in microarray because of multiple problems. One problem is the saturation kinetics. The other problem is that the hybridization efficiency of all the pairs is not identical. So sometimes you might get an overestimation or underestimation of abundance simply because of more or less efficiency in hybridization. So these are the two reasons why microarray is not very quantitative. So if you want to, after getting a hint from microarray, if you want to get quantitative estimation, their quantitative RT-PCR is a very good method for a follow-up.
So, sir, as you said that after doing the microarray, we can validate our results by doing in situ RNA hybridization.
Yeah, but you have to understand the context though. Like as someone pointed out, that if you are studying cancer, it is unlikely that you will go for RNA in situ hybridization. If you are studying developmental biology, yes, because where like you are, most of your questions will be trying to understand molecular difference between neighboring cell populations. There, RNA in situ hybridization is a more appropriate one. But if you are trying to establish that, look, say, like different grades of a cancer, let's say prostate cancer, okay? Say prostate cancer has 6 different grades, okay? I'm making up all these numbers. And you want to identify molecular markers for each of these stages. Okay. Then you do microarray and you find genes that are differentially expressed in grade 1 versus grade 2 versus. Please try to understand, I am not talking about any causation analysis. I am only talking about association analysis. That known grade 1 tumor, known grade 2 tumor, known grade 3 tumor, and so on and so forth. You just figured out what are the mRNAs that are differentially expressed. Now, to verify that indeed they are differentially expressed, then of course qRT-PCR will be a very good method. Right?
So sir, even in situ RNA hybridization, we will get only a qualitative estimate.
Yes, only a qualitative estimate, yes. Because that is also a precipitation reaction, right?
Yeah.
You only get qualitative. Like we make some very weak, stupid attempt to say that, you know, this is more expressed than the other one because the signal intensity here is much stronger than the other. Those are vague scientific arguments, but even I write it. But those are bad arguments. Anything else?
No, sir.
Okay, I see, I have discovered that there is a student I have never seen in the class. Hoimi Banerjee. She has my same last name and she is hiding. Hoimi, please prakat hoiye aap. Where is Hoimi? Shreya, did you raise hand again?
No, sir, I forgot to lower the hand.
अच्छा. होइमी को ढूंढो ग्रुप में. Paul बोलो.
Sir, good evening, sir. I have a question in RNA in situ hybridization. Sir, especially I couldn't understand the role of secondary antibody because while identifying the gene of interest of mRNA, we have the complementary probe which is fluorescently labeled with antibody. Then, so if that goes and binds to our gene of interest mRNA, then the role of secondary what is the role of secondary antibody over there? We can is it the same with southern blot? How it is happening?
No, there is no secondary antibody in RNA in situ hybridization. Let me draw it for you. So this is your mRNA, okay? Now,
yes, sir.
This is your RNA in situ hybridization probe that is hybridized, okay?
Okay, sir.
Probe, you have deoxygenin modification, right? So
yes, sir.
there are some dig labeled CTP. Okay?
Okay, sir.
Now you will use an antibody that is anti-deoxygenin coupled with alkaline phosphatase. Okay?
Okay, sir.
So that will bind like this. And with all alkaline phosphatase is attached. Right?
Yes,
now,
sir.
If you add NBT BCIP here, then it will form a purple color precipitate right here, right? And that's what you will be detecting. So there is no secondary antibody. There is only one antibody, anti-deoxygenin coupled with AP, alkaline phosphatase.
Okay, sir. Sir, is there any criteria to the design? Is there any specific length for that complementary probe, which is we are making it up?
No, I have worked with as small as 200 nucleotide. And in some extraordinary circumstances, like if you read the latest paper from Subimal Sindhu in Professor Sen's lab, he has used successfully even 60 nucleotide long probe. It is very, very strange and weird that what works and what does not work. Like when you make the transcript in vitro, then we typically run it on the gel to see how much transcript is made. For some strange reason, one of my favorite genes called Sonic Hedgehog, nobody has ever seen the transcript on the gel, but it gives outstanding RNA in situ hybridization staining. So the length, the amount, there is a lot of that we still do not completely understand. And we hand wave and say that, it is hybridization efficiency that matters.
Okay, sir.
Anything else?
Sir, actually in lectures, you have told about the secondary antibody.
Yeah, yes, sir.
You have
Yes,
told
sir.
about
You have
the
told that
anti-go.
first there will be anti-ditch, it will be the goat,
Yes.
and then we have secondary antibody, that is
No.
antibody.
So then I made a mistake. It does not matter, but in this specific case nowadays we used directly conjugated antibody. We no longer use secondary antibody.
Okay.
Sir, this portion only I did not understood well.
Okay, let me check once. Okay, before what happened is that these are all technologies that you do in the lab and then and I am now so old that I have not done any experiment in the lab for last 14 years and बुढ़ापे के कारण मेमोरी भी काम नहीं करता है.
Hello, sir. Sir, if you use secondary antibody, then you can enhance our fluorescence signals.
Further. Yeah, you can do that. But
Yes,
let me quickly see what is the standard one. I think there is for fluorescent in situ hybridization, maybe we do not use, and for regular RNA in situ hybridization, maybe we use. Just let me check once, okay, before I give you a definitive answer. It's better that I check and tell you. Let me show you where in the paper you get to see this, okay? You can see my paper, right?
Yes, sir.
Yes.
Okay, let me see how. So in papers, there will be a section on materials and methods. Okay. Oh. You can go there. No, there is no secondary antibody. You see here? So I misspoke there. Okay. The bound probe was detected with alkaline phosphatase conjugated anti-Dij antibody. Okay.
Okay, sir.
Okay, I misspoke there.
Okay.
Like not that it makes a huge difference, but the normal method is that we use only directly this. So someone asked this question, is this question still needed, Ayush? Will you briefly define how do we find out unique 60-mer sequence? Ayush?
Sir, hello.
Uh-huh.
Sir, actually, it was not very clear to me. Also, your voice was breaking in between, so I could not
Okay.
get it very clearly.
So, like as I said that next generation sequencing is today's microarray techniques, these are post genome sequence techniques. Okay. So today you know the sequence of every single mRNA, right?
Yes, sir.
So you can potentially write a code to massively align all the mRNAs against each other, right?
Yes, sir. Yes, sir.
And then you can ask to identify a stretch of 60 nucleotide that will not be present in any of the other mRNAs.
Okay.
So that's how it is done.
So that we can do by doing blast as well. Like
Exactly,
we can.
exactly,
Right.
exactly. It is just that these companies, they have conducted this massive scale blast to generate. So these became their IP, intellectual property. That
Yes, sir.
this probe sets, etc. Yeah.
Okay. Thank you so much, sir.
Mm-hmm. Yeah, Shubhajit.
Yes, sir. Sir, in this, in the RNA in situ hybridization, is the RNA in situ hybridization is also done with the specific 60-mer probe?
No. RNA in situ hybridization is done the way we learned yesterday, that you just clone the cDNA. You do not care if it is not the full length cDNA, and then you just make the antisense RNA in vitro and you do the in situ hybridization.
So then in RNA in situ hybridization, we are not going to ensure the different homologs of any gene.
Actually, we do. And the way we do is this. Let me explain that to you. We actually do. We care a lot about it. So, can you see my blank slide again?
Yes.
Okay. So, it is fairly well known now that even if two genes are very similar, you can do this yourself, okay? BMP2 and BMP4, okay? It is known that the homology will be more in the coding region, okay? The sequence similarity drops dramatically in the untranslated region. And if you remember how we are making the cDNA is by conducting RT-PCR, right? So you have you use your oligo-DT primer to make the first strand, right? So whatever cDNA is being made, whether any other stretch of the mRNA is present in there or not, the 3' most UTR will always be there, right?
Yes, sir. 3' UTR will always be.
So
because this is the first sequence.
Yeah. So these are therefore 3' UTR biased clones. So if we have to let me show you something. Just hold on, let me see if I can find it. Just give me a minute. Can you see it?
Yes, sir.
Yes, sir.
So this is my own data. So in a limb bud, I was trying to detect BMP2 and BMP4. Okay, they are very similar to each other. Not only that, I was also trying to show that I have knocked out BMP2 and BMP4. Or can you see my cursor?
Yes, sir.
That I have knocked it out only in here, but left it untouched here. So only in here I have knocked out BMP4, but left it untouched here. So these were the two objectives. So for that, I had to make probes that will not recognize each other. So I made probes only from the 3' UTR region of BMP2 and 3' UTR region of BMP4. They are unique to each other. Okay, so you can see
they will not have any sequence homology between them.
Yeah, between the 3' UTR BMP2 and 3' UTR of BMP4. It is generally taken as the operating principle that the sequence homology will be more in the coding region than in the untranslated region or non-coding region of a gene. Because you understand the selection pressure, evolutionary pressure is more on the coding region. There, if an amino acid is changed, it will lose the activity. But in the 3' UTR region or 5' UTR region, if some nucleotide changes, it may or may not dramatically affect the function. So it will be tolerated more. Do you understand what I'm trying to say?
Yes, sir.
So the evolutionary pressure ensures that the coding region is more conserved. So now I could explain it, Shubhajit?
Yes, sir.
Okay. Saurabh, Sandarbh, bolo. Sandarbh, you had a question? Okay.
Am I audible, sir?
Yes, you are audible.
Yeah. Sir, you had mentioned in one of your slides that methylation can't be done in vitro reverse transcription.
Yeah.
I think you had explained it, but I didn't understand the reason.
So
why can't I methylate?
Because then you will have to add the methylase as well. Like like I will give you an example. Just a minute.
Sir, I'm unable to hear you.
I'm not saying anything, so you cannot hear me. Just give me a minute, huh? Okay. Can you see my screen?
Yes, sir. Yes, sir.
So this is a like a good thing you are also learning about this technology. This is called site-directed mutagenesis technique. Okay. So say you have a clone. Yeah. Hello.
Sir, sorry to interrupt, but your video froze and the internet disconnected, so I couldn't get any of these things.
Okay, I'm saying it all over again. I have not started. It is not my video. I think your video froze. Who is this? Sandarbh?
Yeah, it's my internet connectivity.
Okay, fine. So I am starting to give the answer now. So this is this method called site-directed mutagenesis. Okay, what is the objective here is to change one amino acid in a clone, in a what if. Okay, the easiest way it is
Yes,
done
sir.
today is you design two primers going in opposite direction. Do you see here? Can you see my cursor? Hello, can you see my cursor?
Yes, sir.
Okay, so these two primers
Yes,
going
sir. Yes,
in
sir.
opposite direction. So now they will make this whole circle, and you will have now the exactly the same plasmid, just one nucleotide or two nucleotides changed here, right? Unless you say
Yeah,
yes, I
okay.
do not understand that you were understanding.
Yes, sir.
Okay,
Yes,
now
sir. I'm saying yes.
you have changed the sequence. Now, if you want to grow this clone, and this clone is already there, so you will have a problem. You will not know which one you are growing, right? So a
Yeah.
cool technique is used. An enzyme called DPN1 is used. DPN1 can only digest the methylated DNA and not the unmethylated DNA. Okay.
Okay.
So as if you read here, it says digest parental methylated and hemimethylated DNA with with DPN1. Why parental one is methylated? Because this came from bacteria.
Okay.
In vivo, it is methylated, while this is a product of PCR reaction.
Mm.
It is not methylated. So DPN1 will digest this and leave this intact. Now you simply transform
Okay.
bacteria, you will grow only the site-directed mutagenized plasmid, which is not methylated. But of course, when you are growing it in the bacteria, then it will get methylated again. Did you get it?
Okay, sir, but like when do we use that DPN? Like,
After you have done the PCR amplification, then
okay.
no, this is just
Okay,
an
I think I get it.
example to show you that in vitro synthesized DNA, they will not have methylation unless you specifically add a methylase from outside to methylate it. But I have never used such a method for my purpose. There are other groups that study epigenetics. Maybe they use it. I do not know. But for normal molecular biology, you never methylate a piece of DNA. Okay.
It can be done, right?
It can be done,
So it can be done,
but I do not know of any molecular biology application of that. Also,
okay.
I do
Okay,
not
sir, one
know.
more thing, like, yeah, please continue, sir, please continue.
I do not know if the fidelity will be 100% because methylase may not work only based on sequence recognition. There might be some other collaborating proteins needed, the context of nucleosome, or in case of bacteria, the bacterial equivalent of nucleosome, those might also be needed. So I do not know how efficient and accurate it will be.
Okay, yeah, okay. Okay, so one more small doubt was there, like, regarding the transposon, I saw somewhere that this kind of small sequence in the DNA which can move from one part to another part. I don't understand how can a part of the DNA can move from one to other part.
So there is a separate topic I am supposed to teach you. So this semester, because of this format, the teaching speed is pretty slow. But if we can pick up the pace, I will talk about it specifically. So what happens is that these transposons, they have a sequence that allows for homologous recombination, and most of the transposons, they have their own reverse transcriptase with it. These are like a kind of remnants of retroviruses. Okay, they have their own reverse transcriptase, so they will make RNA, and then from there they will make DNA, and that will go and get somewhere else.
Okay, thank you, sir.
Okay, fine. Is it all done?
Yes, sir. Sir, one question. Sir,
Okay.
I have read at some place that RNA microarray, it is used to study epigenetic status of the genome, but I don't understood how my microarray, how microarray is used in studying epigenetics.
Yeah, how microarray is studied, used to study epigenetics, right? I also do not know. I have never studied that. If you want to specifically learn about it, I can try to, quickly study it. Epigenetics is something that is never on my radar.
So like
Loves that topic.
epigenetics mainly is all about, you know, the long non-coding RNAs and all those. So it revolves around RNA.
No. There is a way to do methylation sequencing, so they can actually accurately predict where the methylation events are in a normal DNA, like in different kinds of DNA. But I really don't know that topic at all. Okay?
Yes.
Yes, sir.
Okay. So no discussion class tomorrow, and all the best. Saturday 9 a.m. we meet. Bright and early. Okay. So study hard. Okay. I will.
The questions like will be asked at the point, and we have to answer at the point.
No, I will give you the question paper 2 hours before, right?
Okay, sir. So 7 o'clock we will get our question paper for the first batch of students.
Yes, and then I will ask you the answers. There I will not give you any time, because I have already given you 2 hours, right?
Yes, sir.
So at that time, I just want the answers fast.
Okay, sir.
Okay. And there will be questions on primer design, etc. Those you should send to me by email or something like that. Okay. But all within that 2 hour. After your Viva is done, no answer from you will be entertained. Okay. So yeah, be careful with that part. Okay. Chalo. Bye.
Okay.
Good night, sir.
Bye bye. Good night. Good night.
Thank you.
We'll see some of you on Saturday. Bye.